Hello students, welcome in biology classes. Now, in the sequence of the structure of chromosome, cell cycle and cell division. Now, in the previous class, we have discussed about the different changes in the cell during the interphase, prophase, metaphase. Now, in the continuation of that, now we are going to discuss about the changes during the anaphase. So let's start with the anaphase. Anaphase. So the anaphase, in case anaphase, now the cell show the following changes. Now inside this metaphase, as we discussed, that is in the metaphase all the chromosomes comes on the equatorial line. Okay. Now in the anaphase, this anaphase also will discuss in the two part, that is the early anaphase and late anaphase. So in the early anaphase, what kind of the changes we have seen? So this is the centriole, this is the elder centriole, that is the two, this is the aster rays, and the spindle fiber. This is the spindle fiber. Now, there are three kinds of spindle fiber as we are going to discuss. So, the spindle fiber, as in the case of the metaphase, all the chromosome comes on the equatorial line. Now, when the anaphase starts, this chromosome starts to split. So if the chromosome is looking like this is the centromere and these are the two chromatids. So these two chromatids, they are connected at the centromere position, and this is the spindle fiber. Now due to shortening of this spindle fiber or a contraction of this spindle fiber, there is a pull or tension is created. So due to this pull or tension, now this chromosome is split from the middle, that is from the centromeric position, and now these chromosome is split into the two part, that is two chromatid. So one chromatid, it moves upward, and the other chromatid, now it's going to move downward. So this is spindle fiber. So in this case, now we will show such type of the situation. So this is the chromosome, and now this chromosome splits into two parts, that is two chromatid. This is one. Now another one is second one. This is second one. This is third one, and this is fourth one. So here we also show this kind of splitting. Okay. So in this condition, what you have seen? This is chromosome number 1. This is the one chromosome. This is the one chromosome, and when it splits, it gives a two part. That is, it changes into two chromosome. Now, if we start with the 4 chromosomes, so in the anaphase, the chromosome number becomes double. So 4 becomes 8. Now here there are three types of the spindle fiber. One spindle fiber which are continuously associated with the pole to pole, that is called polar. So three types of spindle fiber. Three types of spindle fiber. The first one is polar or continuous fiber. So it connects from pole to pole. The second, on which the chromosome get attached. So this is called chromosomal spindle fiber, or we can call it discontinuous spindle fiber. And the third one, that is between the two chromosomes. When the chromosome splits and moves far apart, this is the part of the spindle fiber, which this is called interzonal spindle fiber. So there are three types of the interzonal spindle fiber. So the chromosomal movement is due to the stretching of this interzonal spindle fiber. So how the chromosome moves toward the opposite pole? So in the stretching of the interzonal spindle fiber and the contraction in discontinuous spindle fiber. So discontinuous spindle fiber, that is जहां पे क्रोमोसोम अटैच है, तो यहाँ पे ये कॉन्ट्रैक्ट करेंगे, यानी कि इसमें थोड़ी सी सिकुड़न आएगी, and this middle one becomes stretched. So when it becomes stretched and this part becomes shorter, then it creates a pull or tension, and that is why this chromatid, or now called chromosome, they moves towards the opposite pole. They are now moves toward this pole and they are moving toward this pole. One more change, that is the plasma membrane. So the plasma membrane now show the depression or pinch. So this is showing the pinching off. So this is a kind of depression, that is cleavage furrow. So the cleavage furrow starts and it is seen from the early anaphase. Now, so what kind of the changes we have seen in the early anaphase? Now the chromosome splits into two parts to give two chromatid, and now these two chromatid now called chromosome. So these are the chromatid and now they are called chromosome. अब इनको हम क्या बोलना शुरू करेंगे? Chromosome. So पहला जो change हुआ, that is the splitting of chromosome. So the first change is, now we will take up this letter on. So first change is splitting of chromosome from centromeric position and into two chromatids now called chromosome. अब इनको हम क्या बोलेंगे? क्रोमोसोम कहेंगे. Okay, so first change is splitting of the chromosome from the central position, and this is called chromatid, and now the chromatid called the chromosome. The second one is due to contraction in the discontinuous fiber. So the chromosome starts to move toward opposite pole due to two reasons. That is contraction. First one is contraction in discontinuous spindle fiber, and the second one is stretching of interzonal spindle fiber, and we also required ATP. So ATP also required. So as you know, there are 36 ATP is required for per chromosomal movement. क्रोमोसोम को मूव कराने के लिए अपोजिट पोल पे कितनी एटीपी की जरूरत होती है? 36. सो हाउ मेनी क्रोमोसोम हियर? दैट इज नंबर 8. 4 upside, 4 downside. So that is 8 chromosome in this situation and 8 into 36. So there are so many ATPs are required for the movement of this kind of situation, this kind of the movement. So first changes splitting of the chromosome from the centroid position into two chromatid, now called chromosome. Now the chromosome starts to move toward the opposite pole due to what? Contraction in the chromosomal fiber or a discontinuous fiber and the stretching of the interzonal fiber. And the third change, that is the cell furrow, the cleavage furrow starts from the early anaphase. So cleavage furrow starts from early anaphase, and due to formation of anaphase, due to formation of contractile ring, and this contractile ring is formed by the actin and myosin protein. Actin and myosin protein. So यहाँ पे अगर हम प्लांट की बात करते तो यहाँ पर जो साइटोकाइनेसिस हो रही है, यानी कि जो डिवीजन हो रहा है सेल का, वो किसकी वजह से हो रहा है प्लांट में? गोल्जी बॉडी की वजह से होता है. लेकिन अगर हम एनिमल केस की बात करें तो यहाँ पर कॉन्ट्रैक्टाइल रिंग का फॉर्मेशन होता है, एंड दिस कॉन्ट्रैक्टाइल रिंग इज फॉर्म बाय द एक्टिन एंड मायोसिन प्रोटीन. सो दिस क्लीवेज फरो स्टार्ट्स टू फॉर्म फ्रॉम द अर्ली एनाफेज एंड इट्स बिकम मोर डीपर एंड डीपर इनसाइड. Okay, so this is the kind of changes which we have seen during the early anaphase. So let's understand what is the changes which we have seen in the late anaphase. Now the next is late anaphase. So early anaphase we have completed. The next one is late anaphase. Late anaphase. So what kind of the changes we have seen in the late anaphase? Now this is the cell membrane, and now the cell membrane becomes, if we talk about the cleavage or a furrow, so this cleavage becomes more deeper from periphery to center. So this is the cleavage furrow becomes more deeper in the late anaphase. So now this chromosome, this is the centriole, this is the asterase, this is the spindle fiber. So this is the spindle fiber, and now the chromosome reaches up to the opposite pole. So these are the chromosome. Now they are finally reaches up to the opposite pole. This is third one and this is fourth one. Okay, and this cleavage furrow, this is the cleavage furrow, becomes more deeper, So this kind of the cell division, this is from periphery to center. तो यहाँ पे जो साइटोकाइनेसिस हो रही है वो कैसी हो रही है? साइटोकाइनेसिस कैसी हो रही है? फ्रॉम सेंटर टू पेरिफेरी, फ्रॉम पेरिफेरी टू सेंटर, फ्रॉम पेरिफेरी टू सेंटर. तो उसको क्या बोलते हैं? सेंट्रीपिटल, सेंटर की तरफ अगर हो रहा है तो सेंट्रीपिटल टाइप, सेंट्रीपिटल टाइप। तो यहां पर देखिए जो क्रोमोसोम है अपने पोल की तरफ पहुंच चुके होते हैं। नाउ दिस क्लीवेज फरो बिकम्स मोर डीपर एंड नाउ द सेल now goes into the telophase. So in the telophase, so this is the what kind of the changes? Chromosome reaches up to the pole and the cleavage furrow becomes more deeper, like a centripetal type from periphery to center, and this is formed by the formation of contractile ring, and this contractile ring is formed by the actin and myosin protein. Okay, now the next phase is telophase. In telophase, what we have seen, so this is the plasma membrane. Now this plasma membrane becomes more deeper, and now this is the situation, so it becomes more deeper, and this is the second longest phase. ये प्रोफेज के बाद सेकेंड लॉन्गेस्ट फेज है। नाउ दिस न्यूक्लियर मेम्ब्रेन रीअसेंबल्ड, प्रोटीन्स जो होते हैं वो रीअसेंबल्ड हो जाते हैं। सो न्यूक्लियर मेम्ब्रेन reappear, nucleolus again reappear. So inside the nucleus there is a nucleolus, so nucleolus reappear. Now the next changes, chromosome, this chromosome now thin out, अभी क्या होंगे, decondensed हो जाएंगे, so it becomes thin out as we have seen in the case of prophase. So this is the chromosome decondensed to form chromatid. Okay, so this is the situation which is just reverse of prophase. So this is the condition just reverse of prophase. Okay, so in the prophase what we have seen, that is nuclear membrane अब डिसैपियर हो रहे हैं, न्यूक्लियोलस डिसैपियर हो रहा है, और जो अब क्रोमोसोम है वो अब कंडेंस्ड होना शुरू हो जाते हैं, और वो अब जो क्रोमैटिन होते हैं वो कंडेंस्ड होना शुरू हो जाते हैं और क्रोमोसोम का शेप ले लेते हैं, यानी क्रोमोसोम के फॉर्म में आ जाते हैं। यहाँ पर एक चेंज और है, दैट इज द शेप ऑफ द क्रोमोसोम। इन द एनाफेज the shape of chromosome becomes more clear, यानी यहाँ पे 3-4 तरह के शेप आपको नजर आएंगे। दैट इज, एक तो आपका हो जाएगा मेटासेंट्रिक, दूसरा हो जाता है सब मेटासेंट्रिक, the third one is acrocentric, and the fourth one is telocentric या telocentric. So metacentric, that is V shape or K shape, submetacentric that is L shape, acrocentric का मतलब होता है J shape में होगा, और telocentric मतलब I shape, यानी कि यहाँ पे P आर्म और Q आर्म की साइज की बात हो रही है। So in the metacentric both the P arm and Q arm are same. In the submetacentric P arm छोटा होगा, Q arm बड़ा होगा। एक्रोसेंट्रिक में पी आर्म बहुत छोटा होगा, क्यू आर्म बहुत ज्यादा बड़ा होगा, जबकि टेलोसेंट्रिक में एक आर्म बिल्कुल गायब हो जाता है, केवल एक क्यू आर्म दिखता है। ठीक है, तो यहाँ पर मैं भी इस शेप की हम बात कर लेंगे। जैसे यहाँ पे आ जाइए, अगर हम मेटासेंट्रिक की बात कर रहे हैं, मेटासेंट्रिक, तो मेटासेंट्रिक में क्या होगा, दिस इज द सेंट्रोमियर, दिस इज द पी आर, this is the q arm, both p arm and q arm are same, p is equal to q, दोनों ही सेम होंगे। अगर हम सब मेटा की बात करेंगे, सब मेटासेंट्रिक, तो सब मेटासेंट्रिक में क्या होगा, p जो होगा वो शॉर्टर होगा एज कंपेयर टू q, तो यहां पे हमें क्या नजर आएगा, एक p arm नजर आएगा और उससे थोड़ा बड़ा q arm नजर आएगा। ठीक है, यहाँ q arm थोड़ा बड़ा नजर आएगा। दिस इज द क्यू आर्म। अगर हम थर्ड सिचुएशन की बात करेंगे, दैट इज एक्रोसेंट्रिक, सो इन एक्रोसेंट्रिक द पी आर्म इज वेरी वेरी शॉर्टर देन क्यू आर्म, तो यहाँ पे पी आर्म जो होगा वो छोटा सा होगा, जबकि क्यू आर्म जो होगा वो काफी बड़ा होगा। दिस इज द थर्ड सिचुएशन। एंड नंबर 4 दैट इज टीलोसेंट्रिक, तो टीलोसेंट्रिक में पी आर्म बिल्कुल गायब हो जाएगा, केवल क्यू आर्म होगा। सो दिस इज द सेंट्रोमियर एंड ओनली वन आर्म, दैट इज द क्यू आर्म। सो दिस इज लाइक अ वी शेप, दिस इज लाइक एल शेप, दिस इज लाइक जे शेप एंड दिस इज लाइक आई शेप। तो यहाँ पे एक एनाफेज में जो क्रोमोसोम है उनके डिफरेंट शेप नजर आने लगते हैं, तो ये आपके 3-4 तरह के शेप हो गए। So this is the final telophase. In the telophase, this is the condition just reverse of the prophase. Now this cleavage becomes more and more deeper. Nuclear membrane reorganized, nucleolus reorganized, and now the chromatin, the chromosome becomes decondensed to form chromatin thread. So the last stage is cytokinesis. So let us see what kind of the changes we have seen in the cytokinesis. So in the cytokinesis, that is after this telophase. So after this telophase, again it is divided into two cell. So one cell, this is the one cell, this is the second cell, that is cytokinesis takes place. This is cytokinesis कैसा होता है, सेंट्रीपिटल टाइप, दैट इज फ्रॉम पेरीफेरी टू सेंटर ड्यू टू कॉन्ट्रैक्टाइल रिंग फॉर्मेशन, ड्यू टू फॉर्मेशन ऑफ contractile ring, and this contractile ring is formed by actin and myosin protein. So this is formed by actin and myosin protein. So due to formation of contractile ring, this cytokinesis takes place. So what kind of the changes? This is the centromere. Again, this is the centromere. This is the complete formation of nuclear membrane. In this, there is again a nuclear membrane. Now, this is the nucleolus. So this is the nucleolus, and this is the chromatin thread. So this is the chromatin thread. Okay. So two daughter cells. This is two daughter cells are formed. So this kind of the changes we have seen in throughout all the phases, that is from the interphase up to the last cytokinesis. So we have seen interphase. After interphase, now the cell enters into the prophase. Prophase has two stages, early prophase and late prophase. From the late prophase, now the cell enter into the metaphase. From metaphase, it enters into the early anaphase, then late anaphase. Then after telophase, and after the telophase, now the cytokinesis finally takes place. That is the cytoplasmic division takes place, and this is of centripetal type and due to formation of contractile ring. Now the two daughter cells are formed. Okay, so this is the cell division and cell cycle, and in the next class we will discuss about the C value, that is the DNA content of the nucleus of the inside the nucleus, and the number of chromosome. So what kind of the changes takes place in the nucleus, that is in the chromosomal number, and also the DNA content. So in the DNA S phase, in the S phase of the interphase, DNA replication takes place. So DNA content will be double. So what kind of the changes which is takes place in the chromosome number and also in the C value. C value, that is the DNA content which is found in the genome, that is in the haploid nucleus. So haploid nucleus में जो DNA content होता है उसी को हम क्या कहते हैं, C value कहते हैं. So we will understand the C value and what kind of the changes in the C value, what kind of the changes in the chromosome number we will understand in the next class. Okay, thank you.